Well, thank you very much, everyone, for joining me for this webinar. As you can hear from my accent, I'm not from America, I'm an Australian, so I hope you'll be able to understand everything which I'm saying throughout this. Sometimes it can be a little bit different to how most people pronounce things in America. What I wanted to do when I got asked to do this webinar, I thought about different options which would be helpful to people in clinical laboratories. And what I struggled with a lot when I first started doing parasitology was the diagnosis of Entamoeba species. So we would get a lot of specimens through in labs, which I worked in hospitals, from particularly refugees, full of Entamoeba. And when you haven't seen a lot, it can be quite difficult to differentiate the species. So in this webinar, I want to go through and explain the different things which you need to look for to be able to differentiate the different species of Entamoeba. We're going to focus a lot on the Entamoeba histolytica complex because it is the one we're most concerned about, and Entamoeba coli because it's the one you're most likely to see after histolytica or with histolytica. But I will also talk about some of the rarer species and show you slides of those species so that you can get some idea of the morphology of them and how to differentiate them from histolytica and coli. So let's move straight along. We've got 45 minutes to talk about and we've got a lot to go through. So here are the Entamoeba species which are found in humans or which have been reported in humans. You'll see there's Natali there in the histolytica complex group. It's not in your slides. It's only ever been reported that I'm aware of once in a zookeeper from Belgium who was working with primates. But it certainly can infect humans and it's morphologically identical to histolytica. So this first group of 5, they're all morphologically identical. We cannot differentiate them by morphology and they're known as the Entamoeba histolytica complex. So there's several species of which only one is histolytica. And that species, Entamoeba histolytica, is a significant human pathogen. It can cause severe disease or death. Many people who are infected with it are asymptomatic. They have no particular symptoms, but if you're the unlucky one who is symptomatic, you can have anything from mild diarrhea through to chronic diarrhea, through to amoebic dysentery, and it can even go into sterile sites and cause things like liver abscesses and lung abscesses, etc. So quite a nasty pathogen which we really need to pick out and let doctors know about when we're doing laboratory diagnosis. Now the others in the group, Bangladeshi has been reported from children in Bangladesh and apparently does cause diarrhea in those children. Moshkovsky has been implicated as a cause of diarrhea as well, but Dispar and Natali, in those where we've seen infections in humans, they appear to be asymptomatic. So that's the Entamoeba histolytica complex. You'll see there's some other species below. Entamoeba coli, hartmanni, Entamoeba polecki, Entamoeba chattoni, and Entamoeba gingivalis. Now these are commensals. Entamoeba coli, so Entamoeba histolytica always has between 1 and 4 nuclei. The others have varying numbers. Entamoeba coli has anything between 1 and 16. Entamoeba hartmanni between 1 and 4. Polecki about 1, sometimes binucleate. Chattoni also 1 or binucleate. And you'll see that Entamoeba polecki and Entamoeba chattoni are another morphologically indistinguishable group. Now there's also Entamoeba gingivalis, which is found in the mouth cavity. The first 8 which are listed there are found in the human intestine primarily and in the gut. Of course, histolytica may cause extraintestinal infection. Entamoeba gingivalis is found in the mouth. It has been very rarely found in the female genitourinary tract when associated with IUDs. Now, there's something called Entamoeba histolytica or the Entamoeba motility hot stool test. And I want to show you what we're looking for there, because when you do these things normally, you don't get to see the motility of the protozoa. Here is film, which I took from a child with amoebic dysentery in West Africa, of an actual Entamoeba histolytica moving in a slide. So this has just been filmed with a camera down a microscope lens, and you'll see this lovely moving trophozoite here. It has a direction of movement. It sticks out pseudopodia and actually moves in a specific direction. It actually has a directional movement. Those pseudopodia are quite long and it's moving quite quickly. Now that, if you don't have access to stained preps or PCR or anything a little bit better, that can be used and can be very helpful in the diagnosis of species, because the other entamoebas don't have such directional movement and they do not move as quickly. So when you're talking about Entamoeba coli, hartmanni, polecki, they tend to be much slower moving, and they tend to also not have a particular direction of movement. Whereas here you're seeing Entamoeba histolytica moving with a distinct direction in the way it is, and quite quickly. I want to point out there are some other things moving in the background there, such as here. This movement you're seeing here in the background, that is Pentatrichomonas hominis. So this person has two parasites. They have both Entamoeba histolytica, which is a pathogen. They also have Pentatrichomonas hominis, which is a commensal. That's very common. People who have protozoa infections tend to have multiple protozoa infections because they've been exposed to fecally contaminated food and water. So particularly in those who have come from developing countries, refugee camps, etc., you almost expect when you see one protozoa to see more. What I want to point out about that is when you're screening for protozoa, if you see one protozoa, don't stop and just report that. Keep looking because you'll find more. Now the key with these motility tests is it's called a hot stool test because you need to get the specimen quickly. If you don't get the specimen within 20 to 30 minutes, these trophozoites start to dissolve and they lose their motility. So you really do need to get this specimen to yourself whilst it's still warm and keep it warm or you won't see the motility. It's helpful in those cases where you do not have access to things like stool ELISA, PCR, or stain preparations. I don't recommend making diagnoses of certainly Entamoeba histolytica complex without doing a stain prep. But in some cases you have no choice if you're in a laboratory which doesn't have those stains, and things like this can be helpful. So let's move back to the slides now, shall we? And we'll go on with the rest of the PowerPoint presentation. Now, when I was at university, I got told that if you saw a trophozoite of an Entamoeba eating red cells, which is known as being a hematophagous trophozoite, it has ingested red cells, such as the ones in this photograph here, then it's Entamoeba histolytica. And I used to go around telling everyone, if you see ingested red cells, you can report it as Entamoeba histolytica sensu stricto, the true species which causes pathogenicity in humans. However, or disease in humans. However, that has since been disproven. You'll find recent papers which have described both Entamoeba histolytica Entamoeba dispar and Entamoeba moshkovsky, which have ingested red cells. They don't eat as many red cells as the histolyticas, but they do ingest red cells, and so I'm no longer confident to make that species diagnosis simply on the presence of ingested red cells in trophozoites, and that's an important new bit of information for us. Let's go through the morphology. So the biggest challenge which you're going to come across in the laboratory is differentiating histolytica from Entamoeba coli. They're the two you're most likely to come across, histolytica complex and Entamoeba coli. Of course, histolytica complex includes the significant pathogen Entamoeba histolytica, and the E. coli is just a commensal. We don't think that that's a problem for patients, so we're not so worried about it. So we must be able to differentiate them. Now, the things we need to look at with this are the size of the cysts and trophozoites. In parasitology, size does matter. So I would highly recommend that if you don't have an ocular micrometer on your parasitology microscope, which has been properly calibrated using a stage micrometer, that you actually go and get one and you put that into your microscope, because you really do need to measure the size, not just of protozoa, but of helminth eggs and things like this. Very, very helpful in making a diagnosis. The next thing I look at when I'm trying to determine what a species of an Entamoeba is the number of nuclei in mature cysts. So there is a pre-cyst stage, a stage where an Entamoeba is just beginning to end cyst, it hasn't reached maturity as a cyst, and sometimes they don't have the total number of nuclei that we normally associate with them, and I'll show you some of them during the course of this presentation. But once a cyst has matured, the number of nuclei in that cyst is very helpful for us in determining what species it may be. The next and probably the most important step is the morphology of the nucleus in cysts and trophozoites. By looking at the morphology of the nucleus, it really is very helpful for us in differentiating the histolytica complex from Entamoeba coli. And finally, if you find them within cysts, chromatoid bodies, which are also known as chromatoid bars, the shape and size of them can help you with differentiating those two species. Now, one of the problems I used to have when I was learning all this was I've just thrown a whole lot of jargon at you. What you just talked about, chromatoid bodies and all these other things, I didn't understand always what these were. So what I want to do now is to go through the actual anatomy of an Entamoeba. So if you've got this printed out, keep the printout with you as we go through the actual microscope slides, which we're going to move on to shortly, because it'll help you as I use terms to know what I'm talking about. So here's a generic Entamoeba cyst. The first thing I'd note is the cyst wall. This is a nice circumscribed cyst wall. These are normally oval or round. The cyst wall is very distinctive. You should be able to see a cyst wall, particularly in wet preps. So if you can't see a distinctive cyst wall in a wet prep in particular, I'd start to be concerned it may not be an Entamoeba cyst. This is the chromatoid body. These are only seen in precysts and cysts. So you don't see them in the trophozoite phase. This large, dark body. This is a glycogen vacuole. If you have a fresh stool specimen that's not preserved, that will stain quite brown in iodine, because glycogen does stain brown in iodine. So you may see a vacuole staining brown in iodine in a fresh stool, but not in the preserved stools, because the vacuole won't take up the iodine in that case. Here we see two nuclei, and cysts tend to be multinucleate depending on the species. So those are your two nuclei. Now, around the edge of the nucleus, there's this dark stuff, and that's called peripheral chromatin. So the shape of the peripheral chromatin and how it's distributed is very important in determining the species of Entamoeba. And in the middle of the nucleus, there's a dot, and that's called the central karyosome, or the karyosome. And that is also important in differentiation of species. So, cyst morphology. Cysts are spherical. They're non-motile because they're in the trophozoites are motile, cysts are not motile. They're in a inert state, but still alive. If we have a look here at the Entamoeba histolytica complex, cysts are on average 12 to 15 micrometers. There's a much wider range of size, but the average size is 12 to 15 micrometers. And cysts generally have 1 to 4 nuclei. So a precyst will have 1 or 2 nuclei, a mature cyst will have 4. You never see uneven numbers of nuclei because the nuclei work by binary fission. So if you're only seeing 3 or 5 nuclei or something like that, you're not actually seeing them all, because there should be 1, 2, or 4. They divide by binary fission, so there's always even numbers. Entamoeba coli, the cysts are larger on average. They average 10 to 35 micrometers. As I say, there's a size range up to about 50, but most of them are between 10 and 35. And they have up to 8 nuclei most of the time. So a mature cyst will have 8 nuclei. It's not impossible for an Entamoeba coli cyst to have 1, 2, or 4 nuclei if it's a pre-cyst stage, so it's not a fully mature cyst. Very occasionally you will see supernucleate Entamoeba coli with 16 cysts, but that's a very unusual observation. Let's talk specifically about the Entamoeba histolytica complex and the morphology of its cysts. So, how do I know that what I have here in this picture is an Entamoeba histolytica complex and not an Entamoeba coli? The first thing is that the peripheral chromatin of the nuclei, so there's 4 nuclei, the peripheral chromatin of the nuclei is evenly dispersed around the edge of that nucleus. It's very even and nicely dispersed. The karyosome, this dot in the middle, is central, and it's quite tight. It's quite small and tight. And finally, the chromatoid bodies are elongate and they have rounded ends. So that's distinctively histolytica. Let's compare that now with E. coli and see the differences. This is a cyst of E. coli. It's a little bit bigger. The peripheral chromatin is uneven. You'll see that it's not evenly distributed, that there are areas where there's no peripheral chromatin and other areas where there's quite big lumps of it. The karyosome is eccentric, so it's not absolutely in the center of the nucleus. And it's a little bit bigger and more diffuse than you would expect to see in an Entamoeba histolytica. Finally, these chromatoid bodies have jagged ends or pointy ends such as this, quite different to those which you see in Entamoeba histolytica complex. So those are the major morphological features of the cysts that differentiate them. Let's look now at the trophozoites. As I said earlier, size matters. So Entamoeba histolytica complex, average size of trophozoites is 12 to 15 micrometers. And the Entamoeba coli, generally the trophozoites are much bigger, 15 to 50. And normally 20 to 25 is the standard range. Trophozoites of Entamoeba only ever have 1 nucleus. In trophozoite stage, no matter what species there will only ever be 1 nucleus in that motile, active trophozoite stage. Let's have a look at the Entamoeba histolytica complex trophozoite. Let's have a look at those nuclei again. The nuclei are the same. The morphology of the nucleus is the same in the trophozoite as it is to the cyst. So this nucleus here has lovely, evenly distributed peripheral chromatin and a tight central karyosome. Important to consider when you're looking at these trophozoites is the cytoplasm. In this case, the cytoplasm has a lot of fine vacuoles. It can be described as having a ground glass or a bubbly vacuolar appearance, but it doesn't tend to ingest bacteria or yeasts. Let's now compare that to Entamoeba coli. Once again, the morphology of the nucleus is identical to that which we see in the cyst. This uneven peripheral chromatin, where in some parts there's not much of it, in other parts there's a lot of it. So it's unevenly distributed. And an eccentric karyosome, which is probably often a little bit more diffuse, a little bit larger and less tight and dark than you'd see in Entamoeba histolytica. The cytoplasm has vacuoles, but they're not nice and evenly sized and distributed, and there's a lot of ingested bacteria in here. Sometimes you'll see ingested yeasts. Colleagues of mine at DPDX, Henry and Blaine Matheson, have actually seen an ingested giardia in a Entamoeba coli. It actually ate a giardia cyst. So strange things happen. This webinar is about morphology, but I think it's important because of the great significance of Entamoeba histolytica sensu stricto, true Entamoeba histolytica, that I briefly discuss how to differentiate that from the other members of the Entamoeba histolytica complex. So as I said, there are 4 you might come across in humans. There's also Entamoeba natale, but it's only been reported once in the Entamoeba histolytica complex. Histolytica is by far and away the most pathogenic and the most important. If you have not performed a test such as a PCR or an ELISA to specifically identify Entamoeba histolytica in your sample as being true Entamoeba histolytica sensu stricto, do not report it as Entamoeba histolytica. Report it as Entamoeba histolytica / Entamoeba dispar. Scientifically, the most correct report would be Entamoeba histolytica complex, but I advise against reporting that on a diagnostic laboratory report. And the reason I advise against it is because it confuses doctors. When they receive a report saying Entamoeba histolytica complex, they may not understand the nuances of what that means and that this is possibly not a true Entamoeba histolytica. The standard process is to report as Entamoeba histolytica / Entamoeba dispar, and the majority of doctors will understand what that means, that you know it looks like Entamoeba histolytica, but it may be one of the other in the complex which are potentially non-pathogenic. If you want to identify, if you see something that looks like an Entamoeba histolytica complex in your patient's fecal specimen and you want to identify it to see if it's truly Entamoeba histolytica, I think the optimal method is an Entamoeba histolytica sensu stricto specific PCR. PCR for feces is getting quite good these days. You can extract feces and get good DNA, and the advice I give is do a PCR specific to E. histolytica. Make sure you have validated your PCR properly and make sure you have a grinding phase before you do the DNA extraction to just break up those cysts. Just grind it for 1 to 3 minutes, break up the cysts, because it can be difficult to extract DNA without breaking them up. There are ELISA kits available which target Gal or GalNAc lectin, and there is another target which is used in one of the ELISA kits available. These are specific to Entamoeba histolytica, so you can do an ELISA, an antigen ELISA on your feces, and if it comes up positive, you know it's E. histolytica. A very important thing to note, not all commercially available ELISAs that detect E. histolytica are specific to E. histolytica sensu stricto. Some ELISAs will report or will become positive with any group of the E. histolytica complex, and many of the rapid tests will become positive with any group of the E. histolytica complex. So you must very carefully check your own test and make sure before you report something as E. histolytica that it truly does, that your ELISA or rapid test truly is specific to E. histolytica and is not going positive with any of the complex, so Dispar, Moshkovsky, etc. If you recover something that looks like Entamoeba histolytica and it comes from a sterile site, so it can cause disease, it can cause liver abscesses, it can cause lung and brain abscesses, it can infect the skin, particularly around the anus of children who haven't had their bottoms wiped properly. It can rarely also, by the way, infect the genital region. So I have seen photographs of genital infection in the penis and in the vagina of females. if you find an Entamoeba histolytica from a sterile site or from a ectopic site from outside of the intestine, an extraintestinal site, you may report that as Entamoeba histolytica sensu stricto, because Entamoeba histolytica is the only species that goes outside of the gastrointestinal tract. And when I say the gastrointestinal tract, I include the mouth. Unlike what I was taught at university, if you see ingested red cells, you cannot report that as Entamoeba histolytica sensu stricto, because we now know that Dispar and Moshkovsky will eat red cells, and so you must still report those as Entamoeba histolytica complex unless you have performed a PCR or an antigen ELISA that is specific for Entamoeba histolytica. So that's all that I had in these slides to talk to you about background. What I want to do for the rest of this webinar is now go through the actual organisms under a microscope. Now, this is the bit which I think hopefully most of you will get the most out of. So here I have a wet prep, and you can see here a lovely Entamoeba cyst. You see the very, very defined round cyst wall. That's very specific. Let me zoom in a little bit. And here's the cyst. Now, what I first do with these in a wet prep is I go through the planes of focus and count the nuclei. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nuclei possibly. That doesn't make sense. It should be 8 to 16. So this may even be a supernucleate cyst with 16. But there's at least 8 nuclei in there, which means it has to be Entamoeba coli. It cannot be any other species if it's got more than 4 nuclei in the cyst. It's difficult to see the morphology of the nuclei here. It's very hard to see that peripheral chromatin and whether the karyosome is central or eccentric, or if the peripheral chromatin is even. But because of the number of nuclei in that cyst, I'm happy that that's an Entamoeba coli. Now, I would not report only Entamoeba coli. I'd like to see more before I make my final report. So here's another one. As I move through the planes of focus, I see 1, 2, 3, 4, 5, 6, 7, 8 nuclei. So that's an Entamoeba coli again, cyst of Entamoeba coli. This is in a saline wet prep here. Keep scanning. Let's see what else we can find. So you can see there's a lot of fecal matter in here, bits and pieces of fecal matter. This is not an Entamoeba because it does not have a perfectly defined cyst wall. We'll keep scanning. And this is looking nice. So we've seen 2 nice Entamoeba coli. Of course, you have to scan through the entire slide and look for other species that may be present of protozoa. Now I've seen 1 protozoa as a commensal. I'm interested to see what else they may have, because they might have other things. Particularly, this shows they've been exposed to fecally contaminated food and water. Here, you might see these quite a lot in your wet preps. This is a pollen grain. However, to one side of this, we have another cyst of Entamoeba coli. 1, 2, 3, 4, 5, 6, at least 6. There should be about 8 nuclei if I looked carefully. You'll notice this isn't perfectly round. This is slightly oval. That's not unusual for cysts of Entamoeba. So I, of course, would look through the entire thing and look for other parasites, but we don't have time for that. So what I'm going to do now is move on to the next in this list. I want to show you these same organisms now with an iodine stain. Now, iodine, or iodine as it's pronounced in the United States by a lot of people, can help with the nuclear morphology, with just identifying the nuclear morphology. I'm just going to pull this into focus now and correct for white. And there we go. So why we use iodine is it can really sometimes bring out the nuclear morphology well. In this case here, it's really stained up the cytoplasm and it's actually made the nuclei more difficult to see. That is a cyst, believe it or not, but not in very good shape. These are all formalin preserved. None of these are actual live fecal samples that I'm looking at here. But you may find iodine helpful in that it can improve the visibility of cysts and trophozoites in a sample and improve the nuclear morphology and access to that nuclear morphology. Now here's an interesting one. This is a much smaller cyst with 4 nuclei. Now, I'm not going to go into too much detail. Well, I will actually. I'll go into detail on what this is now. What I want to do now is I want to measure it. The most important thing for me now is to know the size of what I'm dealing with. So let's measure this and just see. So this is 12 micrometers, which puts it into just within the size for Entamoeba histolytica. It has 4 nuclei. You probably can't see very well here. Let me delete that and zoom in for you. You possibly may not be able to see very well, but let's see if we can find the actual nuclear morphology a little bit. Very difficult to see the nuclei. The nuclear morphology. And as I've got closer, I see there's actually more than 4 cysts. So this is an Entamoeba coli. It's just a very small one, and that can happen. So this is all Entamoeba coli. Let's move on to Entamoeba histolytica in wet preps. Now I'm on low power here, and something I want to show to you about, this is on ×10, that you can often see on ×10 the cysts, because they're quite distinctive. You see here these refractile round bodies, they're the cysts. They're quite distinctive even on low power, though I don't recommend screening on low power. Now we're down to ×40 again, and let's get this into view. And there we go. Let's now go hunting for those cysts that we saw earlier. So this particular specimen here contains Entamoeba histolytica. That looks like a trophozoite of Entamoeba histolytica to me. Let's measure it. 12.6 micrometers, thereabouts, which is, or 12.5, which is within the normal range for Entamoeba histolytica trophozoites. Let's up that and zoom in a little bit for you, and we'll try and have a close look at this trophozoite in this wet prep. Okay, so you can see there's a nice distinctive border. There's one nucleus, but it's very hard to see anything more of that trophozoite. Let's see if we can find something a little bit better. Here's an interesting thing. This is an early cyst. It's probably a pre-cyst. It seems to only have one nucleus, so a pre-cyst, but there's a chromatoid bar present in that cyst. Now, one of the things which the early parasitologists used to say was it's harder to see the nuclear morphology of Entamoeba histolytica in a wet prep than it is to see of E. coli, and I've found that as well, that when I've worked where I've only had access to wet preps, when someone had amoebic dysentery, you really often couldn't see the nuclear morphology very well, and yet with E. coli's I could always see it very well. Here's another, probably trophozoite, and here's 2 vacuoles present within that trophozoite. But as you can see, it's not easy. It's really not easy to make the differentiation of the entamoebas in wet prep, and I highly recommend that you do not do that unless you have no other choice. What we really need to be doing is permanent stained preparations. This is because it makes viewing the morphology of the nucleus so much easier, and you also have a permanent record that you can show to others or send off to somewhere like DPDX at CDC for us to actually review and confirm your diagnosis. So let's now have a look at trichrome. Most of what I'm going to show you are trichrome stains, because that's the most commonly used stain in the United States. I have used a lot of iron hematoxylin, and I'm quite fond of it. But for the purpose of this, we're going to show you trichrome stains. So I'm just going to bring this into focus now and correct for white, and then we're going to move on. Let's just do that correction. Okay, and now we're going to have a look at this particular slide here. So this is a trichrome stain. What you'll see here is all these lovely bacteria staining up red and blue and green. It's a very pretty stain. I'm going to move down to a slightly lower magnification, might make it easier for you to see things. There we go. And let's now move around and see what we can find in here. Now in this part of the stain you'll see a lot of Entamoeba. They're all cysts, but they've crunched up a little bit and they've stained red, which is actually unusual for this kind of stain. Obviously this particular stain wasn't tremendous, but there are some things I want to show, and in other parts of the stain they are not red, and I'll show you what they should normally look like. But there's a very important point to be made about these cysts. In the staining process, these cysts have started to just scrunch up. Now that tends to happen when you don't bring them to water well, when you don't take them through enough changes of alcohol before you go to xylene. Where I could make a mistake, and this is a mistake a lot of people make, is I measure this cyst to find size and I say, there we go, 14 micrometers. That is the true size of that cyst, is 14 micrometers. If I had only measured the cyst itself here, I would have said it was 11 micrometers. And that is not the true size because it's shrunken. So a very important trap for new players. Don't measure the size of the cyst when they've shrunk. Measure the actual zone or the actual halo around that shrunken cyst. So let's have a look at some nice, lovely cysts in here and have a look at their morphology and describe for you what we can see. I'm trying to find some here which haven't actually got stained red. So here we have a nice cyst. I'm just going to move it to the center for you. Oh. I've lost it, and we'll bring it back. Okay. We'll try and just zoom in a little bit on that. And here we have a cyst. Now here's the nucleus. Bit difficult to see the full nuclear morphology, but you can hopefully see that there's 1, 2, 3, 4 nuclei. Lovely even peripheral chromatin, and most of them have central karyosomes. However, this one here has an eccentric karyosome, and that's not completely unusual. You will occasionally come across coli which have a central karyosome and histolytica with an eccentric karyosome. They don't always read the books, so just be careful of that. Here's some nice chromatoid bars. Let's find another one in here. Now, as I said, whenever you're trying to work out what something is, I mean, there's a lot in this one, but don't forget to measure. Here's another lovely one for you to have a look at. Oh, forgive me, it's a bit difficult sometimes with the microscope trying to get these things in the right spot. Here's a nice example of a evenly distributed peripheral chromatin around the edge here. A nice central karyosome. This one's become a little bit diffuse. Now that's probably because of the thing hasn't been preserved immediately. If you don't preserve your entamoebas soon after passage, the trophozoites start to break down. And one of the first things to happen is that that karyosome becomes much more diffuse and wide and looks a bit more like an E. coli karyosome in that when you have really freshly preserved samples, they have very lovely, tight, small karyosomes. So that's another thing which you can come across as an issue. So what I do is I just look through lots of these things until I've made a decision based on the morphology of many cysts and trophozoites about what I am looking at. Now keep in mind that pre-cysts have about 1 nucleus, trophs always have 1 nucleus, and fully developed cysts have 4. This is a lovely round cigar-shaped chromatoid body in this pre-cyst with only 1 nucleus, central karyosome, evenly distributed peripheral chromatin. Let's measure the size. I'm doing exactly what I've told everyone else not to do. I'm measuring the size there when it's actually, if we measure the size of the halo, we get about 14 micrometers, which is within that normal range for Entamoeba histolytica. So I'm happy that's a pre-cyst with 1 nucleus of Entamoeba histolytica. Let's move on to E. coli. Actually, I beg your pardon, we're going to have a look at trophozoites of Entamoeba histolytica before I move on to E. coli. So here I have a slide which is full of lovely trophozoites of Entamoeba histolytica. I'm just going to zoom in and get it into focus. And here we go. So that's nice, and let's move it back down to standard size focus for you and correct for the white background. And away we go. So here is a very, very nice trophozoite. Just really in nice shape. You see the evenly distributed peripheral chromatin, central karyosome, and look at that cytoplasm. It's quite even and ground glass in appearance. Let's measure the size, and we get somewhere in the region of 17 micrometers, which is normal for an Entamoeba histolytica. Okay, I'm just going to maximize the view here so that you can see more as I go through. Still moving through this trichrome stained here. This is an interesting thing. This is a pre-cyst, so it's only got one nucleus. I've seen a lot of these, and I used to wonder what was going on with my stain, if I'd made a mistake or something had happened in the preservative for this to happen. This is simply, it's just recently become a cyst, and it's still got a large glycogen vacuole in the center. So this is actually a recently encysted trophozoite or pre-cyst. And they do have this situation where the glycogen vacuole can push the nucleus to one side. Occasionally you'll see binucleate ones. That's just something which happens in that early stage, and that'll go away. So when you see that, don't get concerned, it's just normal. Here's another very nice trophozoite as I move through the planes of focus, nice evenly distributed peripheral chromatin, almost central karyosome, not quite central, but as I said, they don't always look exactly as you would like them to. Here's a nice trophozoite that's actually been moving in this direction, and here's the nucleus again, evenly distributed peripheral chromatin, almost central karyosome. So there's some nice trophozoites of Entamoeba histolytica. Now we will look at Entamoeba coli. Okay, so here we are with our slide looking for Entamoeba coli. This is once again a trichrome stained preparation, and here's a lovely cyst of Entamoeba coli. I'll just try and get that more central for you and turn up the light so you can see it a little bit better. So here, nice round cyst, beautiful cyst wall here, and here we have a very eccentric karyosome. You can see it's off to one side of the nucleus, and this peripheral chromatin is not evenly distributed. It's kind of really sort of massed around this pole of the nucleus, and then on this side there's far less peripheral chromatin. Let's have a look at some of the other nuclei. Eccentric karyosome, although quite evenly distributed peripheral chromatin on that one. Uneven peripheral chromatin to one side, relatively central karyosome. And as we move through, we'll just see if we can see any other nuclei easily there. So note that you do really have to have a look at all of the nuclei, you have to have a look at all of the cysts. You can't just rely on, I've seen one nucleus and it's got central karyosome, therefore that is Entamoeba histolytica. You really have to look at all the nuclei, get a picture of everything in the stool specimen before you make your final decision. Something very important, please, if you cannot make a proper species diagnosis, if it's too difficult because it's a poor specimen or you just can't find enough to make it, call it Entamoeba species. It's much better to report to a doctor Entamoeba species unable to speciate than to report something as histolytica which is not, or almost worse, report something as a E. coli or another commensal when it's actually a histolytica and then the patient isn't treated properly for a histolytica infection. Here is another cyst of Entamoeba coli, 4, 5, 6, 7, 8 nuclei. And you'll see uneven peripheral chromatin. You have to kind of imagine a little bit, but I think it's a bit heavier there. Quite eccentric karyosome on that particular nucleus. The nuclei are quite small compared to the size of the cyst as well in many cases. Here's a lovely one in that it's got a very distinctive, this is a real E. coli looking nucleus here with very patchy peripheral chromatin and an eccentric karyosome. So if I go on to higher power with this one and just try and show you, so see the chromatin isn't even and your karyosome is eccentric. So that's it, cysts of Entamoeba coli. Let's move on now to trophozoites. So I'm just going to once again focus my microscope and then bring it back into focus for you. Here we go. Okay, and down to 100, and we'll maximize this view for you all to see on the screen. So now this is, once again, trichrome stained feces. This contains trophozoites and cysts of Entamoeba coli. Let's see what we can see in here. So you have to sometimes search a little bit to find what you're looking for. If you're lucky, they'll have lots of trophs present, but sometimes there are very few. Here's a nice one here. What can I see? Well, I see the karyosome is somewhat eccentric. The peripheral chromatin looks quite even, but it may be slightly blotchy. You can see here there's a bit more there than there is on that part there, but eccentric karyosome, blotchy, bit of stuff in that cytoplasm, which is the ingested bacteria. Let's try and find another. Remember, I never call it off one particular trophozoite. Here, this one trophozoite here is quite a poorly maintained morphology of its nucleus, and I don't think we can really make a call on that one. So that's okay, we keep moving till we find a better one. I never make a call, as I said, off a single trophozoite. I like to see multiple trophozoites before I'm happy to make a call about a species of Entamoeba. Occasionally you may have to, but it's really not advisable if you can avoid it. Here's another nice one here. So, eccentric, very eccentric karyosome, and slightly uneven peripheral chromatin, much heavier there than it is there. So once again, I'm thinking this is Entamoeba coli. Keep on moving, looking. I would, of course, measure these where I could as I went. If I was at all unsure about the morphology, I could use measurement to assist me in making a final decision. Here we have another one. I'm just going to turn up the light a little bit for you. And I will also zoom in to some degree. Very eccentric karyosome. And you can see there's sort of a bumpiness to the peripheral chromatin, although it's quite even. But look in here, see these bacteria? 1, 2, 3, all these ingested bacteria tell me this is E. coli. And if I measure it, it's 15 micrometers. That's within the normal range for E. coli. So those are trophozoites of Entamoeba coli, quite different to those of histolytica, particularly in that nuclear morphology. Let's show you something else here. Here's the third most common Entamoeba of humans that you're going to come across in specimens. So let me just focus my microscope. I'm looking at all these stain preps under times 1000 magnification. I like to look at them under times 1000. If you've got a times 600, you might screen with that and then zoom in on times 1000. But this is what I tend to use for my routine purpose. Now, what is this here? This is interesting. Here I have a trophozoite of an Entamoeba. I'm going to take it down to the normal size to make it a little bit easier, but it looks quite small. It seems to have ingested some bacteria, but it's got what appears to be, it's not in great shape, but it's got a possibly even peripheral chromatin and slightly eccentric, in this case, nucleus. It's a bit strange looking. Let's measure it, shall we? Oh dear. Somewhere between around 8.5 micrometers in size. That's not normal. Remember that I told you that Entamoeba histolytica is 12 to 15, and really the lowest it tends to go in size is 10. So this looks a bit like an Entamoeba histolytica, or even a bit funny, but it's too small to be Entamoeba histolytica. So those of you who are connoisseurs of the Entamoeba will know that this is actually an Entamoeba hartmanni. Morphologically almost identical to Entamoeba histolytica, though they do ingest bacteria sometimes, but smaller. So Entamoeba hartmanni is on average 6 to 8 micrometers. It can vary from 5 to 10 micrometers, but normally 6 to 8, and looks like an Entamoeba histolytica. So let's see if we can find some more in here. These are mainly trophozoites in this slide, which we have, but I'm going to show you some cysts shortly. I used to see this a lot in refugees from Sudan and Sierra Leone. So hartmanni is quite common in sub-Saharan Africa. It's found throughout the world, of course, but quite common in sub-Saharan Africa. Just trying to find, here's a few more here. We'll just move this one more into focus. And you can see there's that lovely histolytica type nucleus, central karyosome, even peripheral chromatin, quite bubbly vacuolar cytoplasm, but only about 6.5 micrometers in size. So that is not a histolytica, that is an Entamoeba hartmanni, and to one side another Entamoeba hartmanni trophozoite. Remembering trophozoites only ever have one nucleus. What I'm going to show you next is a slide stained with an iron hematoxylin stain. I'm fond of modified iron hematoxylin, but it's not popular here. So we in DPDX use trichrome. Why I'm showing you this is a few things. It shows a lot of different protozoa, and it can help. I hope it'll help you see just how difficult it can sometimes be when you have multiple protozoa. And also there are cysts of Entamoeba hartmanni in here. So you can see all of these protozoa here. There's a lot of them. And they're quite shrunken, unfortunately, but this, if we measure the halo, is 8 micrometers. And you can see within it this rounded chromatoid bodies. Those are actually cysts of Entamoeba hartmanni. Now there's a lot of chromatoid bodies in here. I'm sorry it's in very poor shape. You can't see it very well. Something I've observed about hartmanni is you tend to see a lot of chromatoid bodies. These very large things here, these are trophozoites of Iodoamoeba buchlei. This is possibly a trophozoite of Endolimax nana, but buchlei and nana are very hard to differentiate in troph stage morphologically. Here is a cyst of Giardia. So just to differentiate and show you that there are a lot of protozoa in this one person's feces, this is one individual, and this is what you will often see, particularly in people who have lived in very poor conditions such as refugee camps for an extended period of time. They will have multiple protozoa in their feces. Here's a lovely Iodoamoeba buchlei trophozoite sending out pseudopodia. And here's another cyst of Entamoeba hartmanni, very small, measuring only about 6 micrometers, but with, once again, those quite rounded chromatoid bodies that's so distinctive of hartmanni. Not the greatest stain, that one, unfortunately, but hopefully it gave you some idea of some of the things you might see in real practice. I've got 2 last slides for you, which are a bit of a treat. So the first one is something which is a pathogen or protozoa that's found in pigs and monkeys. Sometimes it's unusual to see it in humans. Though it can be found worldwide in humans, it's an unusual finding in humans. Now, the place where it is found a lot is in Papua New Guinea. It's 30% of people in some surveys in Papua New Guinea have this particular Entamoeba, but elsewhere it tends to be considered to be a rare zoonotic event. Now, this here is a Blastocystis hominis in trichrome stain. So just so that you can see what a blastocystis looks like. So that's another protozoa, very common in the developed world. The most common parasite of people in the developed world is Blastocystis hominis. But in here is another Entamoeba, very interesting looking one. I'll just move it more centrally for you. What on earth is that? Now that's an interesting looking thing, isn't it? Big nucleus, takes up almost 1/3 of this cyst. A uninucleate cyst, and the cysts of these particular Entamoebas are normally uninucleate and very occasionally binucleate. It has quite blotchy peripheral chromatin and an eccentric but quite large karyosome, and a lot of chromatoid bars of funny shapes, lots of funny sickle shapes, and a lot of those chromatoid bars. Now, when I hear that, I immediately think of one particular organism, and that is Entamoeba polecki. So this is Entamoeba, correctly should be called Entamoeba polecki / chattoni, because we can't differentiate them, polecki in pigs, chattoni in monkeys. However, it's quite sufficient to report them as Entamoeba polecki-like, and that will be sufficient for people to understand what they're dealing with. Look at the size of that nucleus compared to what appears to be a trophozoite here. Very big nucleus. It's really relatively even peripheral chromatin, but there are areas where it's a bit polar. So it's somewhere in between a histolytica and a coli, and I always think of the nuclear morphology as being somewhere in between a histolytica and a coli, but bigger. And a central karyosome, but quite a large, diffuse central karyosome. Ingested bacteria, seen here in the trophozoites. Cytoplasm. Here we have probably a vacuole here. This probably is one of, there's a funny round body seen in Entamoeba polecki trophs and cysts on occasion. We don't know what it is, but these are mainly vacuoles. I'm not sure if that's actually what that is. This looks to me to be a trophozoite. Once again, very, very big nucleus, evenly distributed peripheral chromatin, slightly eccentric but quite large karyosome. But that size of that nucleus is very helpful. And if we find cysts around the place, you tend to find those cysts, as I said, are very full of chromatoid bodies, And those chromatoid bodies are all sorts of bizarre shapes, lots of different shapes. Here's another trophozoite of Entamoeba polecki. Large, nice large nucleus. I just want to see if I can find one more cyst for you to show you just that mass of chromatoid bodies that you see in cysts and the very funny sickle shapes and pointed shapes of the chromatoid bodies within the cysts of Entamoeba polecki / chattoni or Entamoeba polecki-like organisms. Here's another, not terribly good one, but you can see there's a lot, they're staining red here, a lot of chromatoid bodies. It's a mess of chromatoid bodies with a single very, very, very large nucleus, almost 1/3 of that cyst. That is classic Entamoeba polecki / Entamoeba chattoni. One last, and this is one which I only have one slide of. It was given to me by a mentor of mine, Professor John Goldschmied from University of Tasmania. Extremely unusual to come across this, probably because we don't look. Now this is normally found in amongst dental plaque. That's where you tend to find it. We don't think it's actually involved in dental disease. We just think that it likes to hang out in the dental plaque and eat the dying tissue. Let's have a look at this. What's this here? Well, this is interesting. So this is a scraping from between the teeth of someone who's got periodontal disease. Well, this looks like an Entamoeba nucleus, doesn't it? It's got uneven peripheral chromatin and a central karyosome off to one side. And here's another one, but these are funny sort of almost sausage shapes. They're not perfectly round. And look, the edge of this cyst isn't perfectly symmetrical, and there's a bit here where it's kind of not even got a cyst wall. So that's a funny looking Entamoeba, isn't it? Have you ever seen an Entamoeba that looks like that? Well, no, you haven't, because it's not an Entamoeba at all. This is a degenerate white cell. They can be easily mistaken for Entamoebas. You see just how similar the nucleus of degenerate white cells are to Entamoebas there. It's really quite shocking. And it's a trap for new players. I've made mistakes with this early on in my career, calling things like that an Entamoeba, and they're not. So don't make that mistake. Now this is interesting. This is an amoeba, and it's ingesting red cells. You can't see a nucleus in this one. This is an unusual Entamoeba because it has no cyst stage. This is the only Entamoeba with no cyst stage that infects humans. Here's a lovely example of it. This is a trichrome strain. And this is something called Entamoeba gingivalis, only found in the oral cavity or extremely rarely in the genitourinary tract of women with IUDs in place. Nice, sort of tight, central, often eccentric karyosome, uneven peripheral chromatin, but not very much peripheral chromatin, ingested red cells, white cells, and sometimes even polymorphonuclear white cells, so they eat a lot of things. Very distinctively, you can see the pseudopodia here. When you get Entamoeba histolytica and hartmanni, the pseudopodia tend to disappear when you preserve them, but that doesn't happen with Entamoeba gingivalis. The pseudopodia are very distinctive even in preserved specimens. So here's another one here. All these ingested red cells. Lovely nucleus. They're uninucleate, only ever have one nucleus because they only have a trophozoite stage. There is no cyst stage, so you'll only ever see one nucleate. Eccentric, tight karyosome, evenly distributed, well, small, slightly uneven peripheral chromatin, and distinctively has these pseudopodia sticking out of it. Let's have a look at just one more before we go. This is a lovely one because it has eaten a white cell. So this is a white cell which it has ingested. There's your nucleus, eccentric karyosome, slightly uneven peripheral chromatin. You can see it's heavier there than it is there. And all these ingested bits, here's your pseudopodia and an ingested white cell. So that is Entamoeba gingivalis, and that's the end of the webinar on the morphology of Entamoeba in humans. I hope you found it useful and got to see some unusual Entamoeba, and I hope anyone who's had difficulty, as I did early on in my career, with the identification and differentiation of Entamoeba has learned a little bit from that. So we're going to open up to questions now. If anyone would like to type questions into the system, Pat will ask me, and I will do my best to answer them for you. If biofire is positive and you see an Entamoeba histolytica dispar on the slide, can you call it histo? Yes, you can. If it's positive by biofire, which is a PCR, you can call it histolytica. I believe biofire is histolytica specific, so you can call it that, absolutely. Just check that there aren't other Entamoebas in there like E. coli, because you may need to report Entamoeba histolytica, sensu stricto, and E. coli. But yes, you can call it histolytica specific. It says here biofire. Yeah, biofire does not require grinding. I don't have enough experience with biofire. I always recommend that you follow the manufacturer's instructions. If it says don't grind, don't grind. Follow the manufacturer's instructions and then you will not have any liability if something goes wrong that's because of the kit. So if you're doing biofire or one of the FDA approved ones, follow the manufacturer's instructions. It's only if you're doing internal that you might work through grinding. Fixative, we use PVA. Ecofix is not too bad as well. I like Ecofix because you can still extract for PCR. If you're going to do modified iron hematoxylin, you really want to use sodium acetate formalin, but as soon as you hit something with formalin, you cannot get DNA out for PCR. It'll cross-link the DNA, and that's a pain in the neck these days because we always like to confirm with PCR if it's something unusual. So I would highly advise Ecofix or PVA. I really like Ecofix, but there are lots of fixatives out there. Any of the fixatives that preserve DNA are good now. E. gingivalis at this point is not considered to be a pathogen. It's considered to simply be an amoeba that hangs around. So someone has asked, is it necessary for physicians to treat E. gingivalis? Most people think it's just an entamoeba that hangs around and eats the decaying cells around the areas of periodontal disease. It is closely associated with periodontal disease, but that is not causative. So at this time, the scientific consensus is the E. gingivalis, correction, is not a pathogen and does not require treatment. If you treat the periodontal disease holistically and get rid of the periodontal disease, the E. gingivalis will go away because it will have nothing left to eat.